So morning, everyone. Thanks for coming here. 11 o'clock in the morning today. It's a great turnout. We are going to have hopefully a lot of fun the next hour and a half. We've got an incredible group of 4 people here to lead this discussion on neuroprosthetics. So without further ado, our first guest is Professor of Biomedical Engineering and Radiology and Director of the Ultrasound and Elasticity Imaging Lab at Columbia here in New York. She's written over 140 articles, serves as an associate editor in IEEE Transactions and Ultrasonics. She focuses on the development of novel imaging techniques for elasticity and using therapeutic ultrasound to access and treat the brain. Please welcome Elisa Kanafagu. Our next participant is Associate Professor with the Center of Neuroscience here at NYU. He's pioneered the study of how populations of neurons communicate to guide behavior. He's also invented a novel brain-machine interface that uses the activity of small groups of neurons to decode intentions and other cognitive processes. He's been awarded a Sloan Research Fellowship, the McKnight Scholar Award, and the National Science Foundation Career Award. Please welcome Bijan Pesaran. Also joining us this morning is a neuroscientist and neural engineer and a Lang Endowed Chair in Physiology at the University of California, San Francisco. His research focuses on how the brain uses sensory feedback to maintain accurate and adaptive movement control. And his lab applies this research to the development of brain-machine interfaces. Together with 2 members of his lab, he was awarded the 2013 annual Brain-Computer Interface Research Award for his work on the development of artificial somatosensory feedback. Please welcome Philip Sabis. And our final participant is an assistant professor in the Institute for Computational Medicine in the Department of Biomedical Engineering at Johns Hopkins University. She was a postdoctoral fellow previously in brain and cognitive sciences at MIT. Her research attempts to understand neuronal patterns in the central nervous system in both health and various diseases. She uses computational modeling techniques to model neuronal behaviors, and her applications span a number of disorders of the human brain, including Parkinson's disease, epilepsy, brain-machine interfaces, pain, and even sleep. Please welcome Sri Sarma. So we're going to first show a brief video of a patient who has had a deep brain stimulation implant. These are things that probably a lot of people have seen on YouTube before or on television. And so if we could show that first video, please.
There we go.
So this is actually a patient of mine that we implanted a system in.
Now this is.
So this is Blair Ford, my neurologist I work with, talking in the background. This patient has essential tremor. It's a disorder of the cerebellum where you can see he cannot control his movement, particularly when he tries an action.
Oop.
And this becomes incredibly disabling. He can't drink from a cup. He can't write. So there's his deep brain stimulator being turned on. He says, oh boy, because he gets a quick sensory jolt when it happens.
Can you take this finger, touch my finger, and then touch your nose? And go back and forth a few times?
And so you can see this is really life-changing in the treatment for patients with essential tremor, Parkinson's disease, things like this. But also this patient, it's interesting to note, this system was implanted a decade ago. So what we're doing now, technically, is very similar to what we were doing a decade ago. And what we're going to really focus on this morning is where do we go from here? How do we advance from here?
Still a little.
And if we could now show the next video, just talking a little bit about what deep brain stimulation actually is, and then we can use that as a lead-in to talk about deep brain stimulation as opposed to neuroprosthetics, which is really our focus this morning. So Sri, maybe you could.
Sure, yeah, so what you see here is this is an invasive implant. You go through a surgery, you get leads, electrodes implanted into a target region of your brain. You see something that looks like a pacemaker. That's called a neurostimulator. There's a voltage-controlled current source sends current through those leads that are sitting under the skin so you can't see it, and it activates a volume of tissue in the vicinity of the tip of the electrode. So the idea of deep brain stimulation is, the brain basically communicates through electrical activity. It's the neural patterns, the electrical patterns in the brain that govern our behaviors. So when you're changing the electrical patterns with deep brain stimulation, the hope is you change behavior for the better. So if somebody has disease like a movement disorder like Parkinson's, which resting tremor, rigidity, bradykinesia, the idea is to activate the right part of the brain at the right time with the right signal to suppress those symptoms. So that's sort of the principle behind the therapy.
So in clinical neuroscience, we refer to that as neuromodulation. We're modulating an existing system of the brain in somebody who has a disease that's causing that system to not work as well. But that's not really a neuroprosthetic. So, Phil, maybe you could tell us a little bit about because it's not a subtle difference. What is a neuroprosthetic?
Sure. So when you think about modulation, basically what you're talking about is a circuit that's sort of off kilter. It's out of whack. It's oscillating at the wrong frequencies, or it's sending the wrong patterns. And what you want to do is somehow try to move it back into the right operating regime, if you will. When we talk about a prosthetic device, usually what we're thinking about is replacing something that's missing, something that's broken, restoring function, replacing a part, a prosthetic heart, right, putting in a new heart, and so on. And so when you think about a neuroprosthetic, what we usually think about is replacing lost function. And there are many examples of neuroprosthetics. The earliest one, of course, is the cochlear implant to replace hearing for those whose cochlea aren't working right. And now there are also FDA approved, and in Europe approved for even longer, retinal implants that do the same sort of thing, that stimulate tissue in the periphery, in the eye or in the ear, in order to replace vision or audition. But these days, sort of the new frontier really is thinking about brain implants. So these are implants that would go into the brain that would replace lost function. And there are a number of examples of that. The one that's probably furthest along is a motor prosthetic. So this is for people, say, for example, who have spinal cord injury or who have ALS, Lou Gehrig's disease, who are paralyzed and can't move their bodies. And then what you could do is you could put a set of electrodes, say, in motor cortex, which is the part of the brain, on the superficial part of the brain up here, that normally activates in order to control limb or muscles, lower limb, upper limb, the rest of our body. And you can read out activity from those neurons and use that to infer what it is that the patient wanted to do, and then use a machine or a computer to control some external device that would replace function. So that could be a robotic limb, it could be a computer interface like a cursor, and it could be a wheelchair, it could be, you may have seen recently in the news, there was some news about stimulating muscles. So it could even be functional electrical stimulation of the muscle to reanimate the patient's own limb. And so when I talk about a motor prosthesis, that's usually what we think about.
So, as you were saying, motor prosthesis development has been around for a while. People for almost 10 years have shown pictures of the ability to move a mechanical or a robotic arm. So there's a lot of development beyond that. So maybe people could take a little chance and turn talking. Bijan, talk a little bit about the importance of multiple areas of the brain and integrating those areas to build an effective prosthesis.
So, the brain's a complex network. It's got a tremendous number of connections that link, widely diverse groups of neurons. And there are a lot of disorders of brain activity which depend on that communication being intact. And even a motor prosthetic is a readout prosthetic. You're taking a signal of intent, I want to move like this, and translating it into some overt movement by some other device. You're tapping into the circuit and pulling the signals out, but I think that it's important to recognize, and people do sort of tackle this problem. In fact, some of Flip's recent work is directly related to this, which is that even when you're reading something out from the brain, there's a lot of activity within other networks within the brain that are critical to the construction of the signal that you read out. So even something as sort of overt as a movement has a lot of internal consequences. So how does the robotic arm feel? Where is the robotic arm right now? Is it ready to move like this exactly when you want to move, as opposed to when you're moving in the future? So planning as opposed to execution. Other cognitive thoughts like decision making, like how does the robotic arm know when you're thinking about making a decision to move in advance of moving. And those components of the activity in the brain are really important to doing a good job of reading out something that seems as simple perhaps as a motor readout. And then when you start getting into those more higher cognitive functions, you start thinking about replacing all of them. So somebody who is unable to maintain a memory of a certain type, or is unable to speak, or communicate clearly, right? So you could think of all kinds of other mental functions which give rise to those abilities that you would then be able to repair, potentially with, or even treat and cure, ultimately, with some kind of a brain interface that works across distributed networks.
So Sri, talk to us a little bit about the importance of being able to model neural networks as we think about building devices for humans.
Right. So, I mean, in the context of neuroprosthetics, I mean, what are we trying to do? We're trying to replace, restore some kind of neural function. But neuroscientists, I mean, spend their lives trying to understand what is that function for a very small part of the brain. So, you know, if you go to the Society for Neuroscience meetings, which there's 30,000 people, each are focused on one small subsystem of a system and so forth, and they are the expert there. But it's very hard to understand not only what is that little subsystem doing, but who's talking to that system, who's that system then talking to, and then really understand how the neurons are communicating. What are they communicating? How are they communicating about external stimuli, about your body, your behavior, and so forth? So there's intelligence in these kinds of neuroprosthetics. And that, I mean, besides the barriers of can you put something into the brain that's going to not have a, you know, rapid immune response that's going to make it, you know, obsolete or just dysfunctional and so forth, the first thing you need to do, one of the things you need to do is understand, well, what are you going to replace it with? Right? If there's a brain region that's been damaged and you're going to put something in there to replace it, what is its function? And how do you code that? That's what neuroscientists basically live and breathe, is to understand brain function. So modeling is a good way to come about trying to answer these kinds of questions. So what is modeling? Modeling is basically describing how neurons fire action potentials and communicate those action potentials to their neighbors through equations, through mathematical equations, differential equations that are generated sometimes through biophysical laws. Sometimes you just gather, you collect data and you build statistical models. But what those models are trying to do is to associate some kind of environmental stimuli to how those neurons are firing. So that once you know that association, that's when you start understanding what it's doing and what its function is. And then you can start thinking about replacing it with algorithms and on a chip, basically.
So most of the work that's been in the popular press and things to date has been mostly about unidirectional devices. So being able to harness the movement system and move an artificial limb. And all of you are talking about taking all this knowledge and making it bidirectional, integrating what the brain does naturally. So maybe we can dive into that a little deeper. And Philip, maybe you could start off and talk about the importance of the sensory feedback in building a device.
Excellent. Okay. So I think there's a video we have that I'd like you to look at. So we're talking about patients with spinal cord injury or ALS. So clearly there's something, this is not a patient who looks normal or intact. So can anybody out there guess, I mean, I know it was a short video with a little Mickey Mouse over at the top of it, but can anyone guess what was wrong with this person? Any guesses?
He lost function of his legs.
Good guess. Lost function of his legs, maybe spinal cord injury, maybe stroke. Any other guesses? Coordination in general, maybe some kind of cerebellar deficit or something. Actually, this person's motor system is totally fine. There's nothing wrong with it. The problem is, what's that? Exactly, he's lost his somatosensory feedback. So this shows you that here's somebody who's motorically fine, but whose ability to feel his own body is impaired, and it looks like he has a motor deficit. So we know that sensory feedback is critical in order to be able to execute proper motor control. Okay, so when you look at the current state of motor neuroprosthetics, there have been some tremendous demonstrations of human patients moving robotic limbs, getting pretty good control of cursors to operate, say, an iPad or something like that. And that's really great. But the level of control that you see from those devices is still quite far from what you'd hope to see in terms of natural behavior. It doesn't look naturalistic. It's still slow. It's still labored. A lot like the kinds of movements that we saw there. And so my lab was thinking that what we really needed was to restore the sense of proprioception. And there are a number of labs working on this problem. And it's a tricky problem for a particular reason. Remember I talked before about cochlear implants and retinal prostheses. And they work really well because there's what we call a topography, which is to say that nearby neurons encode similar things, and there's a map. So if you look at the cochlea, for example, it's a nice spiral shape. And as you move along the spiral, you change frequency of the tone that you hear. So if you stimulate here, you might hear a 1 kilohertz tone. And if you stimulate here, you might hear a 5 kilohertz tone. And that's a nice map that allows you to create sets of tones that you know the person will hear and will make sense. But in some areas of the brain, and actually in many areas of the brain, certainly the kinds of areas that Bijan was talking about related to higher cognition, we don't have that kind of topography. And unfortunately, that's even true for proprioceptive feedback, for this ability, when I say proprioception, what I mean is this ability to know where your body is, the kind of things that allows me to do this or this with my eyes closed, because I can feel where my body is. And there's no good, simple map in the brain for that. So when we stimulate, we're going to excite neurons that maybe encode this direction and this direction and this direction, and we don't know what the person will feel. That's a problem. And so here we rely on the kinds of models that Sri was just talking about. We had been thinking about this problem of how it is that you know where your body is and how you combine information. And we had built some models earlier showing that the brain, in principle, could quite readily learn to incorporate new signals, brand new signals that it never experienced before, as long as those signals correlated moment by moment with things that it did know. In other words, that two signals were telling it about the same thing. That was really just theoretical work done by Joe Macon and Matt Fellows and Ben Dichter in my lab. And then Maria Dotterlot and I said, well, maybe we could use this to develop a new approach to prosthetics. So we trained a monkey to do, this is just kind of a proof of concept experiment I'm going to show you. We trained a monkey to do kind of a strange task, which I think there's a video for, where a monkey was asked to make reaching movements. And what you can see there is, this is what the monkey saw. And that little small circle is a cursor that represents where the monkey's hand was on a table. So his job was just to move from one target to the next. But what you can see is he can't actually see where those targets are. Instead, what he sees is a bunch of dots moving along the screen. And he knows that if the dots are moving down and to the left, for example, the target's going to be down and to the left. And then if he overshoots, the dots change direction. So it's this kind of weird online feedback of what he's supposed to be doing. And the reason we set this up this way is it allowed us to create a case where we have an intact monkey, so it's not sort of really a neuroprosthetic application. He's moving his limb. But there's a sensory signal that without it, he can't do the task. And that's knowing where the target is relative to where he is right now. So we trained him to do this task with this kind of weird visual signal. And then what we did in the next video is that we started combining that visual signal that he'd been trained to use with something totally novel. So what you're going to see is on the left there, this is just the same as what we saw before. But on the right, there are 8 lines of little green ticks. And each of those little green ticks is a precise little moment of stimulation, a little pulse of current that we put into the brain that excited a couple of maybe 10 or 100 neurons nearby each individual electrode. We had 8 electrodes, and each electrode was arbitrarily assigned a direction. So for example, in this case, the monkey had to move up and to the right. And so the electrodes that correspond to up and to the right, that have those little red arrows up and to the right, are the ones that are pulsing the highest, and the other ones are pulsing the lowest. And when the monkey first felt this, it probably just felt like some weird tickling on his arm. It didn't mean anything to him. But what we're doing here isn't trying to recreate natural patterns of activity like you would do in the cochlea. But instead, what we're trying to do is harness natural modes of learning in the brain. And the idea is that when the brain sees two things that correlate, that are telling it the same thing, it can't help but integrate. And so it's seeing the vision that it knows, and it's getting this weird pattern of stimulation that it doesn't. And when we first start stimulating, actually, his performance with those green ticks, with that stimulation, is worse than when he just can see the visual cue. But within a day or so, it's about the same. And over the next days and weeks, the performance starts getting better and better and better when he can see those green ticks with vision compared to when he can't, until ultimately what you just saw in that video was there's no visual cue at all. He's sitting in the dark, and he knows what to do because he can feel where the target is from that set of stimulating electrodes. So the point of this experiment was that we can write information into the brain that the brain doesn't know anything about, but we're harnessing the natural modes of activity, which is that the brain is always looking for correlations, looking to combine things that it can make sense of, and in this case using vision and this artificial stimulation. And we think that this is readily adaptable to the problem of providing proprioception to a real BMI, and that's work that's going on in the lab right now, led by Joey O'Doherty, a postdoc in my lab.
So the training effect is really important.
That's right, training is
So
critical.
if you walk, chew gum while texting and going down the subway stairs, over time you'll get better of all four together.
Yes, you will. And you'll annoy a lot of people in the process.
Bijan, so your thoughts on making things bidirectional and harnessing the brain's needs?
Well, so what Flip hasn't shown yet, but I'm pretty positive you're working on this, is looking at the activity in the brain simultaneously to the time when he's stimulating. Right, and so that is technically complicated, but not impossible at all. And then what would be interesting to think about is how he could alter how he's stimulating based on the measured changes in the brain activity that he's recording, either at the same location or perhaps even in other locations. And so now what you're starting to do is bridge the network effectively and alter, and this is the critical concept, altering how you feed signals into the brain instead of being blind to the activity in the brain. Saying the activity in the brain is almost maybe what I would like it to be, to allow someone to perceive the feeling of movement in a certain way, but it would be better if I were to stimulate less perhaps, or in a more precise way. And so combining the recordings with the simulation, specifically at different points in the brain, is I think really important to making naturalistic performance and then extending to those other cases.
So, but all of these approaches, they require recording the brain invasively. So, Sri, how much can we advance modeling without actually having to go into the brain?
So, I mean, there are people who have looked at non-invasive techniques and for neuroprosthetics. I just want to make one point, and it's related to this in response to what Flip has said about harnessing the natural learning mechanisms that are already in the brain. There are heated debates within the scientific community in BMI to say, they're the modelers who are really trying to understand the details of what the neurons are doing, what should be the optimal pattern to get the best behavior that I need, and let me try to put that into the brain if something's damaged, versus other people saying, you don't need to do that. The brain will rewire and figure it out. Just put the implant in, create a very simple mapping, a computer algorithm that's trying to map the neural activity that you're feeding out to some kind of control of a device. And even if it's coarse and not right and not really what the neurons are doing, the neurons will figure out how to use that device to create the desired behavior. So you don't need to do all your fancy math and models. The brain will rewire and do it themselves. So we go through these heated debates on that. But in terms of non-invasive, I mean, it really depends on what your objective is. If you're just trying to, say if you're in a wheelchair and you just want to learn how to control the wheelchair with just natural thoughts, or maybe if the person cannot control the wheelchair themselves, but they want to move around, you could even take something like scalp EEG. People have demonstrated this, where you just put a cap on and you map the signals coming from the scalp EEG into some computer. The computer interprets that and decides, okay, move the wheelchair up or down, turn to the right or turn to the left. Something very simple like that's already been done. So, so it depends on what you're trying to achieve. If you're trying to achieve, fine control of, 23 degrees of freedoms of your fingers and your wrist and so forth, and you want to feel things, it's not clear to me today that we can do that non-invasively.
So let's switch tacks a little bit then. Elisa, you've been working on using ultrasound as a way to access the brain without having to go into the brain. So talk to us a little about the concepts of that, how you do it, what you can learn from it, where we can apply it.
Yes, it's something very counterintuitive. So using ultrasound to either stimulate the brain or change the blood-brain barrier to uptake drugs. So it's something that it's not as intuitive as electrical to electrical. So you put an electrode in the brain and then you can stimulate the brain or modulate it by having it fire actual potential. So in this case, we actually use mechanical perturbations. And it turns out that just as the brain is capable of adjusting the electrical side of the stimulation, we also can do this mechanically. So the brain is actually pulsing at every heartbeat. It's uptaking oxygen by the blood vessels, so it knows what to do with mechanical perturbation. So in the ultrasound case, we can focus through the skull. So that's something that has come about over the past decade. So we figured out the technology through intact scalps or skull. So we actually penetrate through the skin, and we can focus within a few millimeters in the brain, and we can change that. So what you see in the video there is actually the white region that's oscillating is the ultrasound beam. We also can, this is the blood vessel in red, and you can see the tight junctions that are oscillating. These are look like bricks on the lining the endothelial walls, and they open up. And what you can see also is the neuron on the left side that's uptaking the drug now as which is in purple, and reaching finally the neuron that you want to treat. In this case, that will be for treatment of Alzheimer's disease or Parkinson's disease or ALS. So trying to reverse these diseases by modulating, in this case, the blood vessel, the actual vasculature to relax and become more permeable, because our brain is very much, very well shielded when it comes to treatment, to drugs, and which is great when you have a normal brain, but when you have a pathological brain, it's actually a huge bottleneck to actually treat it. So without these microbubbles that you saw there, we can actually also stimulate the neurons, and this is something that has been coming about over the past 5 years or so. And we now can go into subcortical regions, in the subthalamic nucleus or hippocampus, and focus mechanical waves, ultrasound waves in this region only, safely. And we're able to actually get very, very localized perturbations of the brain, and that converts to actual potentials. And what we hope to do is also map the circuitry completely non-invasively. So now when you take the electrodes and you actually go, one region by region, you need, thousands of electrodes to do that. So potentially, we could also go cortically, subcortically. The good thing about ultrasound is that you can actually penetrate over several centimeters, very similar to what you do in imaging. So these are the advantages that ultrasound can offer, complementary to what happens in the electrical side.
So the thought would be to use pulses of ultrasound to stimulate activity or inhibit areas of the brain to get the result you want.
Exactly, yes.
So do we have any idea how ultrasound is actually working to do this?
Well, we have an idea. We don't know exactly the mechanism. So there's definitely the ion channels that are involved. So we have channels on the membranes of the neurons that are actually releasing calcium or potassium or sodium. So that turns out that can be modulated through ultrasound. But that's a theory that some people have actually done with models, and it does check out. But we have to go to the brain and figure that out. It's not easy, obviously, because you can also do this on a petri dish, but it doesn't necessarily translate to in vivo, which the networks are so much more complex. And also the other thing is that we think there's also a net force that from the propagation of the waves is a net force in the entire brain, and that alters the stretch aspect ratio of the neurons. And we know that when you do that, if you take neurons on a matrix and you stretch it, they're going to start firing actual potentials. So both of them may be happening at the same time. The ion channels may be regulated through these mechanical forces, and then we can either inhibit or excite the neurons that way.
So say we figure out exactly how this all works, and then we want to couple it into a neuroprosthesis. So how much can you miniaturize it? How much can you make it applicable to people in everyday life?
We're actually working on that. So there are these transducers that send ultrasound waves and also receive them. So obviously in this case we want to send waves. You can make them as thin as a sheet of paper. It turns out there are materials where you can have these piezo composites, which are piezoelectric. So that means that you have an electric potential that's fired across the crystal that you use, and then that mechanically vibrates at the ultrasound frequency that you need it to. So you don't have to have very clunky big transducers. You can actually make them as thin as a patch. So the idea is actually to put that on the scalp itself. and have some wiring, but in this case, it's actually all external, and have that, having that pulsed at the frequency that you want, for example, the essential tremor to be treated, or in the case of the blood-brain barrier, have it opened when the drug is actually administered IV. So you can have that also replaced if it causes any, infection on the skin, for example. But we can miniaturize that, pretty nicely, and to very, very thin. And also the skull has the curvature that allows it to be serving as an acoustic lens. So it's concave. So when you have the sheet on the concave part of the skull, it actually focuses it well in the region. So you can utilize that as well.
And does the brain attenuate its response over time? If you keep hitting with ultrasound, does it keep working?
So that's a question that we have to answer, but we expect it to be very similar to electrical stimulation. So as long as you're not damaging the region. You can also damage that region if you have to. There is a group at the University of Virginia who does essential tremor ablation, that they take the array, in that case in the MRI, and perform a very small lesion on the order of 1 millimeter in the middle of the brain, and stop the essential tremors within 20 minutes. And it's very, very efficient. So if you wanted to cause permanent, inhibitory activity, you can do that. But you can also do this at lower levels, lower intensity levels, where you don't cause any permanent damage, and you can repeatedly have the same effect.
So we talked a lot about how to harness the brain signal and take advantage of the different aspects to it. Bijan, you've done work trying to think about how do we develop a better end product to then translate through. So talk about some of the issues about, so once you understand proprioception in the brain and the different areas feeding in, how do we develop a prosthesis that can take advantage of that information?
So I think that one of the major issues is that the non-invasive approach is the one which has the greatest market. It's the one that's going to benefit anybody basically who's willing to use the device. It's not dangerous in any meaningful sense, but it's also very limited in its scope. I think this is generally true. And in contrast, if you go into the brain and you get access to all of the relevant neurons, you're going to be able to replace almost any function the brain is capable of. But that's highly invasive, and so that's not very practical and certainly not safe in many cases, or even possible in many ways. So how do you bridge the fact that the technology doesn't necessarily work so well or very effective, it's not that effective, but broadly applicable, but it is broadly applicable in one situation, non-invasive situation, and then when you want to actually achieve the sort of the promise of everything that you could imagine being possible, it's just not practical. And so this is a huge issue because I think it's holding back the field effectively, because either it's impossible or it's not necessarily so what I want to be studying or what I want to be able to give other people. And so the point is that there's another way. And so the other way that we've been exploring is looking across the different scales. So I think what's really critical to think about is the size of the brain. Our brains are about 1.4 kilograms. They're maybe this big, and they contain elements which are neurons, which are about, let's say, 10 microns, so 10 millionths of a meter in size. So you can see a neuron, as you can see something that's 10 microns in size with a microscope that's not that powerful. You can see a group of neurons together. 80 microns is like the width of a human hair. You can definitely see that with a regular unaided eye. And so the brain is made up of billions of these small elements that comprise a 1.4 kilogram whole. And so the point is that if you're looking at a centimeter scale or a millimeter scale, you're looking the surface, for example, at how precise, I'm not familiar with the latest in this area, how precisely can you manipulate neural activity non-invasively at a certain location?
Yeah.
Any general location, not a specific one, but in general. Let's say, I don't know what the answer is. Let's call it 5 millimeters? Less?
It's a millimeter.
A millimeter.
Exactly.
Okay. So within that millimeter, though, there are going to be thousands and thousands of neurons. And those neurons have different connections, and they have different jobs to do, perhaps. And if you could, but still, a millimeter is pretty good. If you could get access to every millimeter separately within the brain, that's a pretty powerful device. It's definitely controlling millions of voxels. And how well that can work is a big question. And so we don't know, because we haven't done I don't believe this has been done in an in vivo behaving situation, something like in a person. But I think that thinking about how well you could do with a millimeter instead of a centimeter, which is typically the resolution of non-invasive electrical devices. So non-invasive electrical devices, EEG system or a brain stimulator of some sort that sits on the scalp. There's a lot of excitement about these. You can go look at neurohacking on YouTube and see some pretty crazy stories that people will tell of stimulating with currents with batteries on the surface of their brain. So that's definitely down to the centimeter scale, basically. And then you want to get down to microns, which is what scientists will tell you definitely you're going to answer the question if you're down at the level of microns. But an engineer would say, well, do I need to be down at the level of microns? This is the big question. And we do not know what the answer is. And I think that it's clear that there's a better way than going a centimeter, which doesn't work necessarily for everybody and do much of interest, or going all the way down to microns, which is optically accessible. You can do this, but this is pretty invasive, totally invasive at this point. And what's in between. And I think that thinking about optimizing interfaces and functionalities of devices for particular cases at the millimeter scale, or my favorite, to be honest, I think you can replace everything probably down to like 100 or 200 microns. I don't think you need to be down at the cellular level, but that's debatable. And maybe a millimeter is just as good.
Okay.
If I could follow up on that.
Yeah.
So I think Bijan laid out the problem quite well. There's a big debate about this, in the field right now as to sort of the scale of operation and how fine of a parcellation we need to be able to understand and manipulate circuits. There's no doubt that we can do neuromodulation with electrodes that are huge. The DBS electrodes are, it's a 2 millimeter,
Yes,
2 millimeter diameter. So these are really big devices compared to the scales that Bijan was just talking about. Yet, when you look at sort of the cutting edge of modern neuroscience in parsing out circuits, what you see is people who are using optical techniques to look at the cellular scale and showing that individual neurons have cell types. Cell type is kind of a very common theme, a buzzword that you hear all the time now in the field, meaning that different cells belong to different classes, and those classes seem to have different functions. And if you manipulate one specific cell type, you might have a very different effect on a circuit than if you manipulate another cell type. And when you think about treatment, so from a scientific perspective, the holy grail would be to be able to manipulate at that cellular resolution across large circuits that span millimeters or centimeters. That's a very tall order, of course. From a clinical perspective, the question is how much of that resolution is needed? And I think that these are still very much open questions. It probably is the case that if you could manipulate a certain cell type at a, say, 100 micron or millimeter resolution, that's kind of a factor of 10 there, that you might potentially have more power than if you could just sort of stimulate all the cells in a local region. And there are a variety of techniques that people are thinking about for this. The most obvious one that comes to mind for people in the field is optogenetics, which is the ability to use light to open specific ion channels that were engineered. So you put these new ion channels using genetic techniques into specific classes of neurons, or into neurons generally. And when you shine light, they open, which causes either a neuron to activate or a neuron to silence. And that kind of resolution, this is now, optogenetics is a huge field. It's sort of taken over in many ways as one of the cutting edge tools in neuroscience. It's pervasive. So that's the kind of thing that is making its way slowly into the clinic now, I think. And there are other techniques. People are looking at ways to do similar sorts of things with electrical stimulation. And there may be other ways, say, for example, not by targeting necessarily using the cellular physiology to target particular cell types, but some of the things that, for example, Bijan is looking at, where you can look at connectivity between two brain areas and target the cells, the projections here, say, for example, of the cells there, which is a kind of specificity that also might offer us a lot of power. So these are kind of all on the frontier, both scientifically and a little bit further on clinically.
Can I follow up with just a quick thing, since we have, I think, some students in the audience? I mean, the technologies are there to basically image small whole brains, right? We can see activity of thousands of neurons across structures. Lots and lots of data is collected, and in certain labs, data are collected all the time. And now, today, we're in a situation where how do we deal with this big neuro data? the typical neuroscientist is not trained to deal with big data, right? These big data scientists, they learn machine learning, optimization, all kinds of pattern recognition techniques. They're good at that, but they don't know anything about the neurobiology and the neuroscience. And that you've got the neuroscientists that are performing these experiments. So I think the next generation, and I think what universities are trying to do, are build training programs to train you guys to be interdisciplinary and to be able to then understand, to Bijan's point, what is it, this single neuron, how does it impact the emergent behavior of the organism? How do you cross all those scales? And then we'll be able to start to figure out, okay, if I want this behavior, I don't need to be at this resolution. I can manipulate up here, okay? And so we still need to understand how things, the dynamics across the scales. And I think where the data's there, basically the knowledge of how to glean that information and model that is still for the next generation, I think, to come and solve.
So that brings up an issue, though, because as Bijan was saying, we've got a big brain. And so we're trying to think about learning really specifically where an area is it's important for a certain task that we want to build a prosthesis for. So there's a conflict there, because we've got all this real estate and we're trying to find a very small area.
Perhaps.
So how do we do that?
I would even question that premise in many cases. I think that the localization paradigm is very influential and important, and there are clearly functions in maps that can be localized to locations on those maps. But I think that those functions are, for the most part, fairly basic or well described through an interface to the external world, and the more abstract interpretive functions of the brain, which underlie our cognitive abilities, are probably not localized to locations, and that perhaps there are functions which can be manipulated by doing a fairly precise knockout or enhancement, because the network will respond in a way which will, lead to the change that we're talking about. Let's be more precise. There could be a complicated network that's responsible for your ability to remember your childhood. But many different parts of the brain, many neurons are involved in this ability to recall your memories. But you may be able to disrupt that by knocking out a particular part of the network, for example, and maybe even restore it by maybe replacing that very small piece. But that may be a specific case of a function where the network itself is supporting the function, but your intervention is having an effect by finding that special piece. It's like the magic bullet in the brain targeting domain. But there may not be magic bullets for a range of things, and I think that the localization dogma is coming under question because it's not being as effective as it maybe should be. And I think that the critical point that you basically can't ignore, this is almost certainly true, is that different brains, our brains are all very different from each other. Every brain is basically a butterfly. And so the challenge of finding that magic spot that you need to interface with to replace a complex disorder, even if it does exist in each brain, you need to find it in each brain. And there's a lot of clinical trials which are targeting complex cognitive functions like depression, mood, obsessive compulsive disorder, all the rest of it, with this localized spot manipulating paradigm, and they look great before they spend the $100 million to do the clinical trial, but when they pick the random subset of people whose brains they haven't mapped fully and then try to intervene, it doesn't work so well because they don't know where to go. So I think there's an inherent tension here. There are functions which could be localized, and the way you treat them is to diagnose it by finding that spot and then going to that localized spot to treat it. Or there could be other functions which are actually not localizable, and you can't actually control the network from a point. And really what you're going to have to do is come up with a more broader distributed description. And so this, I think, this is all basic science debates, but it has very practical implications for the development of technology.
So how do we study this? How do we take the steps forward? Because like you say, everybody in the room is an individual. And so how do you all as a group envision studying humanity in a way where we leverage big data and the variability between brains to say whether or not there's a sweet spot? Is there an endogenous rhythm that we should be tapping into? How do you see going forward to take it to the next step?
Well, I think that, I guess I would say that it depends on the problem that you're trying to solve. So for some problems, I do actually think that relying on learning, as Sri and I were talking about earlier, is probably the way to go. In other words, if you're trying to provide artificial proprioception, it probably would be better. You'd get faster learning and maybe even ultimately a little better performance if you could tap into just the right parts of the brain with sort of the best approximation to the natural code that you could. But if you get close enough in terms of the location of the brain, it'll probably be almost as good. And you could probably stimulate almost anywhere. And a person could learn to use that feedback for control, just like we learn to use all kinds of strange artificial feedback to drive our movements in video games or in driving a car or something like that. And so I think that that's a case where while there may be fine localization, we can probably get away with approximation to that. But there are other problems where I think that this sort of drive to individualization is going to have to be the answer. And I think there are a couple of examples of that. There have been studies, for example, this is just to give you an idea of the problem, in visual cortex, where you can ask, if I'm looking at a neuron right here in visual cortex that might represent some little part of my visual field and some set of features of the visual world or something like that, and ask, who does it connect to, this cell? You might think, well, it connects to its neighbors strongly, and then its next door neighbors a little bit less, and so on down the line. But actually what you see is there's a patchy connectivity. And the connectivity probably makes some sense based on the maps about orientation and other things, but it's patchy, and you can't predict in advance exactly what that connectivity pattern is going to be. And I think that kind of patchiness of the brain, where there are these very small groups of cells that might be like microcolumns is the term that people use in cortex, or just little collections of cells in other brain areas, might be working together functionally, but they're hard to find. This gets to the kind of thing that Bijan was talking about. And when you look at, say, for example, the clinical trials for depression, which haven't worked as well as one would have hoped from the preliminary data, but they do work in some subjects very well, patients very well, as we were talking about earlier before we came on stage. Why is that? And one possibility might be, well, there are just some subjects that are amenable to that form of neuromodulation and some that aren't, and that's a possibility. But another possibility is exactly this problem of trying to find just those little microdomains that are the right ones for that patient. And what do you do about that? I mean, it may be that non-invasive pre-surgical imaging will get to the point that it's good enough that you can find those domains. And that's one of the things that Bijan is using extensively in trying to map out connectivity between brain areas in advance of surgical procedures. And actually, we have a procedure on Monday using some of the same techniques that Bijan is using in one of our animals. But another technique, I think, is to develop neural interfaces that are very flexible. So to be able to safely put in thousands of electrodes, even though you don't need thousands, but you put in thousands because it's safe and it's easy to do, and then you can individualize the treatment after the fact because you've got the bandwidth to do that. And that's an approach that I think a number of people are thinking about as a way to go, and it's something that we're actively working on in my lab, an engineer in my lab, Tim Hansen, who's a very talented engineer working with some engineers at Berkeley, to develop a scheme where we can put 2,000-3,000 very fine, flexible electrodes into a monkey brain in the first instance, of course, actually a rat brain in the first instance. But the goal is to be able to put in so many that we can try to find those microdomains after the fact.
So what's the spatial constraint there? Those 2,000-3,000 electrodes are going over what area of brain?
Well, what we're trying to do is to make it that you can individualize it. So you can put them as close as you want, up to, say, maybe on the order of 50 to 100 microns close to each other. So that's, to think about Bijan's description before, that's maybe about 5 cell width apart, or 10 cell width apart. That's at one end. Or because each one can be put anywhere you want, if you wanted, you could put them with a great deal of less density, but across dozens of brain areas. We think that's an interesting space to work in that hasn't been very well explored yet.
So I think the concept here, if I could emphasize it, is and this is standard in the treatment paradigm, is diagnosis first and then treatment. Right? This is always how this works. Every doctor has to diagnose a disorder before they can properly treat it, and that this plays a very important, critical role in brain treatments with devices. Is that the diagnosis phase itself needs a special device, in my mind, and I think people are doing this, needs a special device which will deal with this variability problem. that sure, you could have a non-invasive stage first, of course, because the diagnosis device itself isn't going everywhere, but it is going more places than you need for treatment. And it will then basically build a model of this brain that's functioning in a certain way that we want to, understand and deal with. And then we will diagnose this person's brain, and then system, not the whole thing, but the functional system that we care about. And then we will either turn off a bunch of the implant functionality to reduce its power load and heat and whatever, or we will do a secondary surgical procedure where we pull out this monster device, so to speak, and then replace it with the targeted device, which may be even endoscopically delivered, maybe through the vascular system, because you get a lot of access to the brain through the vasculature. And then you could have a much less morbid outcome, and yet still have that ultimate goal of precise and effective.
Yeah, for diagnosis, it's important to obviously have, the imaging across scales. So we have, EEG, fMRI, we have MRI, diffusion tensor imaging, and, other techniques that can be complementary at different scales, which would be optical techniques or, more on the electrical side. Those you have to basically probably go with the treatment itself, because diagnostically right now we need more techniques that can go to cellular level, but unfortunately the physics catches up with us, especially in imaging, where you want to have a resolution of 10 microns, but unfortunately that cannot happen over several centimeters. So if a brain was as small as a mouse brain, then we could do the optical techniques that people do, or remove the skull completely and then do diagnostics and basic science. Unfortunately, we know more about the rodent brain than we know about the primate brain, except for what happens in monkeys. But we have this trade-off between resolution and penetration. And we can't really beat the physics, but we can probably go across scales and combine everything that we do here. But you have to have the new generation, like Sri said, of students which are going to be talking in all these languages across scales, because optical techniques are great for microns, electro techniques are also great for microns, and then as you go higher, magnetic, acoustic, even chemical techniques, or even more on the nuclear side, those are higher scales. So you have to understand the brain, you have to understand across scales, and that's going to take a lot.
So you guys are talking about the ability to put 1,000 electrodes in a relatively small space and sample and see what's best. Sri, you talked about machine learning and big data sets. And in the experiment that you showed, Philip, it took a while. It took a while for that to work. How can we use machine learning to speed up the process so that we have an implant? How can we use computer learning to do what we want the patient to do that might take 6 months, so we can ramp it up and basically, like you said before, take advantage of the brain's own hardware and chemistry to let the brain learn itself rather than having to put humans through tasks every day?
Right. Well, machine learning, it's basically the field of, if you have a large data set about some underlying phenomena or process, how can you just take all that big data and uncover sort of the structure in it? And the structure can be much sparser than the data you have. And typically the data is noisy, it's messy, there's missing data, and so forth. So what machine learning is all about is how do you develop techniques and algorithms that are going to find that underlying structure that's much sparser than the data itself, that's going to teach you something about the process. So this is kind of a high-level description of machine learning. So when it comes to big neurodata, you can imagine we are seeing firing patterns of activities of whether it's individual cells or populations of cells and so forth. All of that activity is big data. It crosses time, space, and so forth. So it's complex data, it's noisy, there's variability because you're getting this from multiple brains and so forth. So the idea there is, okay, maybe we collected this data under some kind of behavior, maybe when the animal was making a choice to, pick this pellet versus that pellet. What happened in the brain? Who's talking to who? Who's really sort of necessary to drive that behavior and allow the animal to make the right choice? Well, machine learning would go in there and say, okay, I'm going to, from all of this time series data across space and time, I'm going to try to figure out exactly what's sort of the fundamental network underlying and governing this behavior, it's going to be just a handful of necessary structures, and then how does those structures talk to each other and communicate to ultimately generate the pattern of activity that's going to make that animal make the right choice, right? So it's trying to uncover sparse structure from very dense data, and that's what machine learning does. But with that said, I don't know if you guys are thinking this when Flip and Bijan were going back and forth and talking about the sort of the ideal of placing thousands of electrodes all in the brain so we can, in a non-invasive way. How many of you thought that sounds completely counter to like an oxymoron? Non-invasive, thousands of
invasive.
Okay, minimally invasive. But I bring this up, and I don't know if Guy, you can shut me down if you don't want to go there. But DBS, as you saw in the animation, that's 2 electrodes going in the brain. Okay, and it can be remarkable in terms of a therapy. How many actually get it? So what would you rather do, pop a pill or get thousands of electrodes minimally invasively going into your brain? And I think this is going to be, once we figure it all out, it's going to be an issue of how are you going to, have the population accept this there. I mean, so I don't know if you want to talk about that.
I think that's a great thing. Let's return to that in 1 second, because Philip had a comment on the data side, and then I think we should absolutely return to this.
I can talk about both of those. So I just want to say one thing about machine learning, which is that if you look at the state of the art of motor neuroprosthetics, many groups that are involved in this are using what you could think of as co-adaptive procedures. In other words, we've talked a lot about how people will adapt to the device, but the device is at the same time using ideas from statistics and machine learning, adapting to what the person is doing or the monkey is doing. And that's another way that machine learning can help. So it's not like you buy a machine, you hook it up to your brain, and you need to learn to use it like a car. It's more like something that's constantly trying to understand what you're doing and refit to you.
So I think actually it's a really good idea to return to that, because a very small percentage of Parkinson's patients actually get deep brain stimulation surgery, a very small percentage of essential tremor patients, because there's a fear factor and there's a real risk. I mean, surgery has risks. And so I actually keep a set of scales in my office, because every surgical decision is risk-benefit. You want it to be like that. And so it brings up the issue of not just how do we make it safe, but also what conditions are appropriate. So, I mean, at the beginning, Philip, you talked about there's certain things that are obvious. So a motor prosthesis for an amputee, somebody who's lost a limb in a war, that's an obvious to think about for somebody, or somebody who has spinal cord injury or ALS, Lou Gehrig's disease, things like that. But when you're talking the risk benefit, we've got to make it incredibly safe. We've got to make sure that the early populations we're studying have an incredibly low complication rate. We've got to make sure that we're picking the right populations of people and the right targeting techniques, because if you're wrong early on and it costs $100 million to run a trial and it fails, then it's going to be really hard to go forward. I mean, these are huge issues. So why don't we tackle some of those? So what do you guys think about, as you start pushing it a little bit, what conditions enter your mind as appropriate to think about for neuroprosthetics?
So let me just say one thing before we get into this debate, which is that we're very fortunate to be in a situation where we've got many layers of legal regulation on what we do. And so it's not as if one of us, or even a surgeon, can just decide, what would be really cool would be to take this person who's got blank and stick 100 electrodes in and see what happens. That's just not how it works. There are many layers. You have to convince what's called the IRB, the Internal Review Board, at your own institution. You have to convince the Food and Drug Administration in the federal government. You have to convince a funding agency to pay for it, and they'll look at the scientific merit of these studies, and so on. And so we're in an environment where it's fun to discuss these things, and we should discuss them early on. But I just want the audience to realize that it's not like we get to make these decisions in an environment like this. There's a lot of due process that goes into these phenomena. So I can continue, or I can let someone else take it.
I just want to say, because we didn't talk about the non-invasive stimulation other than ultrasound, which is basically TMS, transcranial magnetic stimulation, or ECT, electroconvulsive therapy. So these are actually used outside the brain, and we still don't know how exactly it works. Sometimes it really works wonderfully, and people are really treated, especially for depression, and it has outstanding effects. But other times, there's no effects at all. And we still have to go back to the basics, understanding how stimulation really works. And we still, so basic to clinical. It has to be a feedback mechanism, constant, going back to the basic, understanding what stimulation does, and then going back and retweaking. And I think that DBS is actually one of the things that works very well, but it is invasive. And it will be nice to actually be able to make it more amenable to patients when it works, and we can learn from DBS and then apply it also to non-invasive or minimally invasive procedures. But I think the loop needs to be constantly closed, and there's a lot of basics that we still don't understand, so that we can.
And we need a lot of advancements in every aspect of technology, right? So, I mean, that DBS system grew out of pacemaker technology. It's 20-year-old technology. We're still using it.
Right.
And so we need to understand what we're treating, how we're treating, and then we need to advance the field of what to actually treat with.
Right. Now, 20-year-old technology might sound like a bad thing. If you're going to go buy a new car, you wouldn't buy a 20-year-old car. So why are you putting a 20-year-old DBS in your head? But there's a good reason for it, because the risk factor of it is relatively low, because it's been put in so many patients. We have a good feeling for what the risks are. And so I think when you go forward and you think about this cost-benefit analysis, the risk-benefit analysis that you were talking about, that, of course, changes over time. Right now, you talked about motor disorders, severe depression, other kind of severe debilitating psychological disorders are the sorts of things that where the alternative to intractable conditions like these, where drugs aren't working, for example, the alternative is a great deal of suffering. Then it makes a lot of sense to think about opening the head and putting in what may be a novel or relatively experimental device. So that's the gain. And then what's the cost? Well, the cost is that every time you open the head, there's a risk. If you put in a device that isn't going to last more than 5 to 10 years, that's an inherent risk right there. And the current devices that we have, not the DBS devices, they may last a little bit longer, but the kinds of more advanced devices that we're talking about for stimulation and recording don't have terribly long lifespans at the moment. So that, I think, will automatically limit where we're going. But I'm actually I have to admit, somewhat bullish on this. I think that as the technology gets better, as we get smaller devices that can be implanted with smaller holes in the cranium, for example, as we get devices that become wireless and that can be wholly implanted in the head with lower power and so on, as we get devices that can stay in the tissue and function for ideally 20, 30, 40 years, the people will see that risk coming down, and as they become more widely accepted, more people have an implant in their head. It won't seem like such a strange and foreign and odd thing. That the scope of conditions that clinicians, patients, and the regulatory agencies start thinking about as plausible and reasonable alternatives will grow. And I think that's going to happen soon. I think on the 15, 20-year time frame, you're going to see a great expansion of the kinds of conditions that people will be getting their head opened to have implants put in to treat.
But this is...
Go ahead.
I just wanted to just bring out the point that this is a very personal choice that is made even in the cases when it's clinically indicated. So forgetting about all kinds of other decisions people make about themselves. And so in the military, for example, they are developing a lot of advanced technologies over the years for prosthetic prostheses. And what's interesting about their experience, this has nothing to do with the brain necessarily, this is just some kind of external robotic system, maybe controlled by the muscles, or maybe controlled by movements of your body in other ways. And as opposed, so the simplest prosthesis you could get is the hook, right? You just mount it onto somebody. Say they're an amputee, they've lost some part of their arm. You can mount the hook onto their arm, and they can use it through some kind of pulley and spring system to manipulate it to hook things with their hook. Right? So this is a very established prosthesis technology. There are more advanced versions which allow the hook to have a hand with 20-some degrees of freedom, or even interface then with the muscles, and then you imagine clenching a muscle and then signals, or you even clench the muscle and the signals go into the now robotic hand and move the hand. So these things basically exist and are available for people who unfortunately need them. And what's interesting about this is that there are basically very different user adoption scenarios. Mm-hmm. That some people will have this implanted or mounted onto them, not even implants in many cases, they're mounted onto their arm stump, let's say, and use it very well and never use it again. And other people who have the same clinical condition will have it mounted on their arm, they'll use it exactly the same way the other person used it, and will use it all the time. And so what divides people into these kinds of groups? And why is it that you get rejection and adoption when you would think that it's all about how long does it last and how difficult it is? These people are using it the same way. They're reporting the same user experience in a sense. It doesn't hurt. It's functional to the same level of dexterity that they could be scored on with some test of performance. And yet there's a totally different decision underlying their use. And so this is true in all technologies, right? So some people can't stand smartphones because they'd much rather have buttons on there rather than, there's all kinds of issues where this is played out in the same basic way. And so I think that it's like any advanced technology, people are going to have a very personal relationship to it, and this is going to, more than I think anything else, ultimately govern this discussion, is do you want to use it?
But just back to your original, is, all the hurdles to get through the process, getting a clinical trial to be more likely to succeed. And I'm not saying this because to give myself purpose, but I think this is where mathematical modeling can play a huge role in the future, which is the idea of virtual clinical trials. What is a virtual clinical trial? Well, if you could model the phenomena, the system that you're trying to change with a drug or with electrical stimulation, if you can model that, you can use predictions from the model to say, yeah, these are the group of people that are going to respond to this very well, but these will not. And this could prevent a lot of failures going down forward. I think the barrier there is the culture of the FDA and then society of just accepting mathematical modeling as, just believing in it, that they can make these predictions and showing that they play a big role in the pipeline going from, idea to clinical trial to actually getting FDA approved and out. Then you deal with the adoption issue.
But I actually think that modeling plays a role in adoption as well. Maybe not so much for the layperson, but for the physician at least, where if after the fact you have a model and you really understand the mechanism and you can show that with that model you can predict maybe how specific subpopulation will respond, or if you tweak the thing in this way, this will change in this way. Even if that model doesn't allow you to improve therapy, what it does is it gives the scientists confidence that they understand why it's working. They can convey that to clinicians, and they can look their patient in the eye and say, we understand how this works. And I think that can help with adoption as well.
So let's push the limits a little. I'm going to give you a hypothetical. So we've got a device that we know. We've figured out how to work the motor system. It's working great. Now we've figured out where a nodal region is that can work on memory. And we've got an MRI system and a genetic test and sequencing together that can tell you if you have a family history that you're going to get Alzheimer's, and it's going to start when you're 60. So you're 59. You know your memory is going to start going away next year. Is it appropriate to think about a neuroprosthesis for that person who's going to turn 60? To help protect their memory as long as you possibly can from the effects of Alzheimer's. How do you guys think about that?
Sure. Yes. I would.
But then if you can fix, you can enhance. And then the young kids will say, well, wait, why can't I get that neuroprosthetic and I'll have enhanced memory and I'll do really well in school and I'll do all kinds of issues come about. But,
well, I think the border between treatment and enhancement is an interesting one. So here's an example. Imagine that you have an implant, well, these exist, for vision, okay? So what that means, right, is that I can't see, and so you're going to give me something where I'm going to have a little camera, say, mounted on my glasses, and it's going to look around the world, and it's going to allow me to see what's around me. That makes a lot of sense, and that's restoration of function, right? Makes sense. But then I say, well, hold on. What I really want to do in the evenings, I want to read, okay? Now, what I could do is I could look at my Kindle, but that introduces a lot of noise. Wouldn't it be better to just have my iPhone talk to the device and put the text directly in? Now, surely that's a good idea. You should do that, right? I think that makes sense. I don't think anyone would have a problem with a blind person being able to read directly from their iPhone. Okay, but if you can do that, then does that mean that I can also use it as a general interface for browsing the web? Well, it still makes sense, right? But then the next person says, oh, wait, I can have my brain talk directly to my iPhone. I want one too. And it's a very, where does that border come? And I think this is where, again, all these layers of, it's not as if I can go buy one at Walgreens and put it in myself, right? These are not going to ever be over-the-counter treatments. And so, I think that, we have lots of examples of treatments where, look at amphetamines as a treatment for ADHD in children, right? It's a real disorder, and it really helps lots of children, and there are lots of people who'd like to get their hands on them. And, sometimes that happens, and bad things can happen, but on the balance, we've got a system for dealing with that. And in some ways, the risk here is less, because it's not like you can get your buddy to give you his brain implant at school. That's not going to work.
So,
I think we have to develop technologies that we can prevent, Alzheimer's or diagnose it early. Because genetic testing, for example, BRCA1, BRCA2 for breast cancer, it's only 5% of the patients. So the fact that you have, the mutation doesn't necessarily, you have a time clock that works that way. It's also stochastic probability-wise. So I think you have the early diagnosis is very key. And I think we're talking about therapeutics here, but I think the diagnosis, understanding, early onset or even preventing the disease down the line, I think these are actually more important, from the point of view of treatment, because, how do you want to treat Alzheimer's? That's the other option. So you have detected it, how are you going to, at what stage do you start? Do you start early? Do you start mid, late? So.
Your enhancement question reminds me of the surgical joke where patient comes in the office and says, well, doctor, will I be able to play the piano well after surgery? And you say, of course you will. We're not going to hurt your pianos. Well, that's great because I couldn't play before. So, we could actually talk all day about this, but I think it'd be really, we've brought up a lot of topics here, and I'm sure some people in the room have questions for some of our speakers about any aspect related to things we've covered or potentially something that we haven't covered. So anyone from the room, questions? In the back over there, yeah. Hold on 1 second so we can get a mic to you, okay, so everyone can hear your question.
Hello, so I'm a computer science grad student actually, and I have this question because since it came out, I'm more interested in learning about the non-localized nature of neurons and how they communicate in general from your perspective, and how does neuroprosthetics deal with that? I mean, just to give you an idea, I think of it like a community on Facebook. Now, just because I have other neighbors to me who are localized, we might not be as well connected, but when you have someone else maybe on the other side of the world, I might be more intimately connected through some other connection, like maybe Harry Potter or something like that, right? So does the same happen in the brain? And if so, how do you guys deal with it in sense?
So, this is the kind of thing that we study in my lab. And we actually had a paper recently where we found that if you just take a group of neurons that you've recorded blindly, let's say, from part of the brain or a couple parts of the brain, and then what you do is you label them according to how well they're working in terms of their firing patterns with other activity in another part of the brain. And so what you can do is you can label them as being sort of communicative, not communicative in a sense. And then what you can do is start asking questions about these now labeled neurons in terms of how well do they predict decisions. So the subjects, all these neurons are actually predicting the decisions that the subject's making at some point in the future perfectly well. So you can predict exactly what this process is going to do later on, 100% no errors. But it takes longer to listen and figure out what's going to be done in the future if you are analyzing only the non-communicative neurons. And if instead you analyze the communicative neurons, you can get that information much faster. And so there already is evidence suggesting that the way you might want to read things out is conditioned on these network properties.
There's a big challenge, though, in what does communicative mean? What's the definition?
That was
you
the
brought up
papers.
that two people are connected because they both love Harry Potter, versus this person lives next door to you. So that's a spatial proximity where you could draw a graph, and that could be, or it could be. So what does it mean to be connected? And there's in neuroscience we have notions of structural connectivity, that this neuron physically projects to the other neuron, therefore it must talk to that neuron, versus these neurons are firing while these neurons are firing during this path. They're functionally connected. So I think these are challenges that we still face, and so yeah.
Yeah, so the whole point was what is our definition of communication is a huge issue. And let's be clear, this is not anatomical connectivity, because I think what's really important to recognize about the operation of these circuits is that they're totally dynamic. Like they are not staying still for more than a few hundred milliseconds at most. And they're constantly updating and shifting around every minute to every hour to every day to every time you fall asleep, you dream, you wake up. So nothing is that stable. And so anatomy is pretty stable on this scale. It's certainly not that stable either. Neurons grow, send out connections, and die. So they also change their anatomical properties. But I think that absolutely this idea of communication is not anatomical, it's functional, and it's highly dynamic. And debatable exactly what it is. But I think that if you can show that a group of neurons that you're labeling as communicative, and then you're predicting what they're saying better, then you've got some meaningful definition of communication.
So you're saying we can all take confidence in the fact that we're fundamentally unstable. Other question? Yeah, over here.
Hello. Very interesting. Oh, all right. Mic's down. Very interesting stuff.
That'd be a microphone prosthesis.
Yeah, I know. It's facing. I'm wondering, you guys are talking about a lot of restorative functions, which is extremely interesting and very applicable. But I'm kind of wondering, once you start delving into that sort of field, how you might start to kind of coax out some more superhuman abilities, certain aspects where you might rewire the brain, or just, I want to quit smoking, I want to lose weight, whatever, and those kind of patterns that emerge in your brain that stop you from doing that. How much might you be able to restore them? Beyond that, how you might even be able to do certain things that are sci-fi, essentially, just beyond the immediate application that we have today and the need to fix your arm doing this, and now you can use it properly and whatever. So I'm kind of wondering if you guys can talk upon that subject.
Superhumanity.
Well, let me just start. Yes. Let me start with the example of smoking, because I think that's a good one, where there's a transient need, hopefully, or any addiction, really. And what about that? And that's a case where I think something like ultrasound, where you could, and that's, I think that may be on the horizon, being able to treat transient disorders with non-invasive brain manipulation. Obviously, we do that already with drugs, but to do it with circuit manipulation like this, I think, is around the corner and is a great idea. And again, it gets to this cost-benefit ratio. What if, once we understood addiction better, you realize that we could do it with stimulation, but it's going to require a massive implant with lots of electrodes? Maybe then no, then nicotine gum is probably a great way to go, or something like that, right? So it depends on the cost-benefit ratio. And I think with enhancement, again, I'm going to kind of just say a little bit more what I said before, which is that I think that although there's no doubt that one could start thinking that way, and although elements of what might look like enhancement will probably be part of the therapies that we use, there will always be a very strict legal and ethical framework, at least in the United States, that runs the show, and that we all have to work in. For now. And for now, that's true. And I think that it would preclude things like getting an implant simply because I want to be stronger than my neighbor.
But that's implants, right? If it's non-invasive. So
Right.
if Lisa continues to
Yeah.
on her successful path,
I'll try.
then how do you prevent
Well,
people
that's
from
happening.
getting I mean, if your kids get some memory enhancement and they're taking the same test my kids are, I want that memory enhancement.
And that's happening.
Right? I
now.
want my
That's
kids to
happening now.
have it. This
There's
is
a
the
company
problem,
in
right?
Silicon Valley that's using TDCS, which is putting direct
Batteries.
current batteries, basically taking a 9-volt battery and
You
putting
can-
it across
you
your
can
head,
actually
seriously,
build it.
to try to get athletes to be able to jump higher. I mean, literally, that's what they're doing. And right now it's not. I guess it's not FDA.
I take the very. I'm going to come back. I come back to this myself all the time. It is a very personal thing, and I think that in ways that are not always obvious. So, for example, let's say that you want to enhance yourself, okay, and you think that there's some problem in a test you can't solve, whatever. Right? So you're going to go ahead and enhance yourself. What you're going to do is you're going to end up changing your brain somehow. That's like the first order statement, otherwise nothing's going to happen. The second order statement then is now your brain is going to adapt. And the brain doesn't know exactly what it is you're trying to do to it because it's got its own, in a sense, mind. And so it is responding to your intervention, which you're only like sort of secondarily aware of. Like your conscious awareness is not a lot of what's going on inside your brain. And so then what you have to deal with is the fact that maybe it's not working very well and you're not actually doing better on the test. So the first thing with all of these implants is that it makes it worse rather than makes it better. So then what you have to do is go, oh, well, I'm going to stop using this. This is the whole adoption thing. I'm going to stop using this because I don't like it very much. Or I'm going to try harder. And then what you have to deal with is improving your interface and using it more and changing it more. And then you basically become very married to your technology. And I think we're all aware of this in our personal lives because we have advanced technologies at our disposal. And I think the basic rule about all of these things is you never use it less. And you always basically use it more. And so I think where this goes is full-blown cyber reality. And this is a choice that ends, because these things don't work that well on their own. This is the whole point. All technology, right? This is becoming my personal philosophy here. But all technology leads to some side effects, which then need more technology to fix. And I think that this is just how it works. And sure, you can get an amazing technology that doesn't require any alteration to anything else. I would like to see a list of those technologies.
I mean, for everybody in the room with kids, try to take your cell phone away from your kid at bedtime, right? We all fight that.
Yeah.
Right? Question? Yeah, in the back.
Thanks. Two things. One, I wondered what of the technologies, ultrasound, et cetera, that you're deep brain stimulation, what are the next practical areas where we're going to see these technologies coming into the clinic? And the second question was just around the interface between digital technologies and pharmacologic technologies. You know, because even in terms of, you know, we now have convection delivery of GDNF for Parkinson's disease to the putamen, et cetera. So I wondered about the way in which drugs will have a potentially a different kind of interface or use in conjunction with these, and as well in terms of digital technologies, a la, you know, Adam Ghazali, and kind of a way in which you can sculpt neural circuits digitally. So it's a lot of questions, but.
This is a big issue, and I mean, obviously we talk about devices a lot here, but there's also the other world, and we alluded to that, the drugs. And there's also another world, which is basically the combination of the two. And that world is very atrophic, and not much has been done there. Why? Because you have the drug, you know, people who are actually experts there, who don't understand devices, and devices, us basically, who don't necessarily understand drugs. So there needs to be another area where the two work together. And in fact, in the brain, we have to make it work, because convection enhanced, for example, that's using GDNF for Parkinson's, that's also, you know, very invasive. It doesn't always work. We don't understand why. These injections that go deep in the brain, 1 out of 10 actually work. There's a lot of, the brain is not actually designed to take up drugs through injection. So there's a lot of engineering of how you do that part. So it's important to actually cross over to the drug world as well, and have devices like you know, the ones that we discussed, and enhance those drugs. But not only the existing drugs, but we can actually design drugs now that can be used optimally in combination with devices. And that's a world that's completely bare right now. I think it's a new frontier. And it will need a lot of efforts to bridge that. And on the order, it's also an order of difficulty at the FDA, because the FDA is also not structured necessarily like this. You have also device versus drug. There is also the combination of the two, but it's very also not very well developed. So I think that's a big challenge. But I think the brain will have to solve that challenge, because we can help the brain from all sides, and we need to use the drug front with the device front together very wisely.
So I think it's a great question. And when you think about treatment of any kind of brain disorder, you think about exactly what you said: drugs, rehabilitation or training, and some kind of circuit stimulation-based mechanism. And in fact, there's one example that's combining those three to great effect right now. In spinal cord injury, there are groups in Southern California and in Switzerland who've had remarkable success with this, and they don't really understand the mechanism, I should tell you, but remarkable success with this three-pronged approach all together. So what they're doing this in rodent, and they're doing it in human patients, is they're using a cocktail of drugs that seems to put the spinal cord, the brain and the spinal cord, into a more plastic state, number one. Number two, they're stimulating to try to, in effect, to try to use plastic mechanisms in the brain that would normally be there to drive reconnection of spinal pathways, while the animal or the person is going through the motion, say on a treadmill, of movement. So there's a little bit of rehab because they're getting the real motion to happen. There's stimulation and there's drugs altogether. And they find, just empirically now, there's no real deep understanding of the mechanism yet. But when you combine those, they get actually quite remarkable effects that are beyond anything I've seen in terms of functional restoration in spinal cord injury. So I think you're absolutely right. Trying to think about how to merge them is great.
So it's neuroplasticity from different directions.
Exactly, that's right. 3 ways of driving that, that's right.
Yeah, another question. Let's go in the back.
Hi, thank you for holding this discussion. So I was wondering, I know other research institutions are also, for example, manufacturing microelectrodes to be really, really small, and then maybe some other institutions are looking at which material is going to be the most biocompatible for these electrodes. So how do you communicate among each other, within each other, to say like, oh, we actually don't need an electrode this tiny, or oh, maybe this material would be better for creating the resonating so that we can create the ultrasound waves. So how do you communicate with these different research institutions so you minimize the efforts of each other's efforts?
So how do you enhance collaboration and communication?
You want to start with?
I think it's a little bit ad hoc still. We could do better there with some data, some data science part. But yeah, so you obviously have to be in touch with all the papers that are across different journals. I would say about 10 journals. You also get your Google alerts and all that stuff. And also in conferences, you have to go and basically have this kind of conversations on a regular basis because it's very difficult. You can read, for example, a journal article and you understand the overall concept and the methods, but there's a lot behind it that's actually never really reported, how many times it didn't work. Actually, nobody reports that, and because it won't get published. So you have to basically guess what happened as you read a paper, how many times they actually did it before it started working. And then you get this nice conversations and the feedback. Unfortunately, it would be nice to have some kind of a database of the electrodes that don't work in specific cases, or the ultrasound that doesn't work in specific cases as well. But that's kind of...
You obsessively ask people the missing question, that the thing that they never made totally public, but underlie everything that basically followed.
I mean,
you just got to find out what that question is.
Yeah, I mean, this whole thing of collaborations, I think the biggest challenges, and fundamentally our academic funding agency, that system is flawed. So if you really want someone to, say, be a neuroscientist and do experiments and really understand how to mine that data, machine learning and so forth, how do you actually train that person? Mm-hmm. Well, that person has to belong to the experimentalist lab. Maybe they're in Flip's lab, and Flip's got some money to do a project. He's got to get that experiment done, because that's what NIH is funding him for. So why would Flip allow his student, or why would he give an incentive to him to send his student off to work with the best machine learning person in his university to really spend some time in that lab when he's paying for that person? And he has to get his stuff done, right? Because he's got the agencies. So we have these issues here, that, barriers that, the agencies are not giving us incentives to train that disciplinary student, to allow them to really understand the device world versus the experimental world versus something else. And I think it's a big challenge.
Actually, I'm going to just give a little shout out to an agency that's funded, I think, everyone on the panel here, which is DARPA, that actually is very good at this sort of thing. DARPA does create teaming. DARPA does let you know when you're on a road that another person that they're funding is on a similar road, and often encourages investigators to talk to each other. And that's something that I think the NIH and the NSF and other funding agencies don't do as much. Ultimately, it's our responsibility to go out and find out what other people are doing, but I think it is something that the agencies could help with.
Yeah.
So unfortunately, we could take questions, actually, I'm sure, for hours and hours, and maybe some of the speakers are willing to individually based on their schedules, but our actually allotted time is up, so we officially have to call it here. And so I'd really like to thank everyone for all their